postdoc on the project. Who was another postdoc on the project and who developed the modeling component of the study. So part of the work I'm presenting today was published last year in Science, where we presented the broad picture, picture on the project. But today I'd like to focus more on the foraminiferal side of the story, which was published just a few months ago in Biogeoscience. And because this is the TMS Cushman Foundation Foraminiferal series, for once I want to really indulge in lots and lots of foraminiferal details. So be warned. And sorry, because I have kind of a little problem with my screen. Okay, so I want to start by just giving a brief overview on how we got the data. It's just going to be just really some highlights, because as I said, it's all published already. So if you want to go more into the details, you just need to go and read the papers. So we worked on a range of globally and latitudinally distributed sites that you see on this map. And we focus on a time window covering the last 15 millions of years. We specifically worked on 7 target ages or time slices between 15 million years ago and modern, because we were interested in capturing these long-term cooling trends of sea surface and deep ocean temperature since the Middle Miocene. So what we did initially was measuring oxygen and carbon stable isotopes on as many species of planktonic foraminifera as possible. And you see here a couple of examples from site 871 and 872 in the tropical Pacific Ocean for the 15 million years old sample and for the modern sample. And as you can see, we really try to capture as many species as possible, including the rarer ones. In terms of preservation, the preservation was generally very good to excellent, as we really carefully selected on which site to work on before ends. Okay, so once we got our foraminiferal oxygen and carbon stable isotope data, what we did was matching the data with oxygen and carbon stable isotope profile from an Earth system model of intermediate complexity, Gini. And each of these profile was obtained for site or area specific and time specific configuration and boundary condition. So the aim of matching the foraminifera and model derived oxygen and carbon stable isotope data was to, from one side, to reconstruct changes in the efficiency of the biological pump through time based on carbon-13, and from another side to reconstruct planktonic foraminifera depth habitat evolution based on oxygen-18. So here I'm showing one of the figures from the Science paper. This is basically the figure that we use to show the planktonic foraminifera depth habitat evolution since the Middle Miocene. I'm showing here 3 representative sites, a mid-latitude site in the Atlantic Ocean, a tropical site in the Indian Ocean, and a tropical site in the Pacific Ocean. The colored symbols here represent deep-dwelling species of planktonic foraminifera living below 200 meters depth. And as you can see, moving from the Middle Miocene to the present, the deep-dwelling species of planktonic foraminifera progressively move down the water column to occupy deeper depth habitat. And new species of deep-dwelling planktonic foraminifera evolve through time. So this was one of the main outcome of the project that we published in Science. However, today I want to follow this story up and specifically try to address these 3 questions. The first question I'd like to address is if there was any other plankton group which were involved by this evolution of deep-dwelling species over the last 50 millions of years, or was it something related to planktonic foraminifera only? Second of all, I'd like to address what could have been the evolutionary drivers behind this depth habitat deepening diversification of deep-dwelling plankton. And then finally, I'd like to dive a little bit into what could have been the evolutionary mechanisms behind the speciation, the origin of new deep-dwelling species in planktonic foraminifera. So to do that, I'm going to use abundance count and biogeographic distribution of deep-dwelling planktonic foraminifera species and abundance count and biogeography of deep-dwelling calcareous nannofossil species. And the calcareous nanofossil data were produced by Amy Jones, who at that time was a PhD student at Birmingham University. Now she graduated. However, she at that time she obtained calcareous nanofossil data from the same sample where I obtained data from planktonic foraminifera. And then eventually, I'm going to compare the abundance data with, again, model outputs from Gini. So to compare planktonic foraminifera and calcareous nanofossil data, what we did was combining species into ecogroup based on depth habitat. For planktonic foraminifera, we used our own paleodepth habitat reconstruction, and we identified two main groups: surface dwelling species with an average depth habitat above 200 meters depth, and deep dwelling species with an average depth habitat below 200 meters depth. And in the deep dwelling species group, we included the majority of the hirsutelids, all the truncorotalids, all the globorotalids, and here with globorotalids, I mean the Globorotalia merotumida tumida lineage, and all the globoconellids. For the calcareous nanofossils, we do not have depth habitat reconstruction. For the grouping, we used Poulton. We used depth habitat from Poulton et al. 2017, who sampled basically coccolithophores at different depths in the Atlantic Ocean and inferred depth habitat based on that. Because our species because the species in our samples are all living today, we could make this kind of direct comparison. So for calcareous nanofossils, we recognize three groups: the upper euphotic group, the lower euphotic, and the sub-euphotic group. Just to give you some example, in the upper euphotic group, we have Reticulum fenestra species. In the lower euphotic group, our representative species is Gephyrocapsa ericksoni, and the sub-euphotic group is largely made by Florisfera profunda. Okay, let's have a look at some data now. Here I'm showing the percentage abundance for the planktonic foraminifera and calcareous nanofossil ecogroups. And as you can see, the deep dwelling ecogroup in planktonic foraminifera gradually rise over time since the middle Miocene, from percentage close to 0 up to almost 20% of the assemblage. For the calcareous nanofossil group, this is the lower euphotic zone ecogroup, and as you can see, it's at almost 0 in the middle Miocene, and then it rises up to almost 80% of the assemblage in the modern. So a huge rise in the abundance of deep dwelling species for calcareous nanofossils as well. In terms of diversity, I'm showing here the Simpson diversity index just as an example, but they basically behave all the same. We have more, but they behave pretty much all the same. The only group which shows an increase in abundance over time is the deep dwelling group in planktonic foraminifera, this bluish line here. All the other lines shows pretty much like they are all pretty much flat lines, except again the lower euphotic zone ecogroup in calcareous nanofossils, which drops in the late Miocene and then stays pretty much flat through time. And this pattern is driven by the evolution of Florisfera profunda in the late Miocene, which becomes so abundant and so dominant, which basically flattens out all the diversity. So we do see basically a consistent pattern in this increasing abundance of deep dwelling species for both planktonic foraminifera and calcareous nanofossils as well. So what could be the evolutionary drivers behind this? And here I'm showing the abundance versus some of our model outputs. The parameters I'm showing here is particulate organic carbon flux extracted for different depths in the water column. You can see here 40 meters, 127 and 228. And oxygen concentration, again, a different depth in the water column. And these parameters are extracted from different latitudinal bands in this work. We distinguish between high latitudes, two different mid-latitudes and low latitudes. Again, if you want to learn more about all the details, you just need to go and read the paper. Everything is explained in great details there. So for now here, I'm just going to say that basically the increase in abundance of deep dwelling species over time basically parallels this increase in POC flux at below the euphotic zone, at about 230 meters depth. And it also mirrors this decrease in POC flux export from the surface. So this parameter down here is basically an indicator of the efficiency of the biological pump. It basically says that there was more of organic matter exported below the euphotic zone in the upper twilight zone. Vice versa, this one up here is basically an indicator of primary productivity, and it basically shows a global decline in primary productivity since the Middle Miocene, according to our model. Oxygen concentration also show big changes. In particular, what we see is an increase in oxygen concentration in the lower euphotic zone and sub-euphotic zone environment, especially in the low latitudes. So it really seems like this increase in deep dwelling species for the calcareous plankton, it has an environmental control. It's driven by environmental changes. And this fits very well with what we showed with the science paper, that basically moving from the warm Middle Miocene ocean to the cold modern ocean, there was an increase in the efficiency of the biological pump because of lower bacterial metabolic rates in the water column, which made organic matter remineralization lower and less efficient, so that more organic matter could penetrate deeper into the water column and basically feed organisms down there, setting up a new deep dwelling food chain. And this makes a lot of sense for planktonic foraminifera, which are heterotrophs. But what about coccolithophores? Aren't they algae? Aren't they supposed to do photosynthesis? So what were they doing down there where there is no light at all? So part of the explanation could come from the global decrease in primary productivity that we see in our model, because if productivity is lower, then surface water tends to be clearer, and therefore more light can penetrate down in the lower euphotic zone and sub-euphotic zone environment. And we think that coccolithophore species adapted to deep water can really maximize the few light that they get to do photosynthesis, can make the most out of it. However, another part of the explanation could come from mixotrophy. So mixotrophy is a type of behavior whereby the organism can switch from being an autotroph to being a heterotroph. And it has been recently shown, like I'm showing here from this paper, that some of coccolithophores can be mixotroph. These two species have been grown for 30 days in total darkness, a different concentration of nutrients. And as you can see, they both grow and reproduce quite efficiently in the absence of any light. So the conclusion of this paper is that they could get nutrients just from the environment through heterotrophy. And so in the absence of light, they can adopt this type of mixotrophic behavior. So if Florisfera profunda could also adopt this type of strategy and switch from being an autotroph when light is available a little bit to being an heterotroph, then this could explain why it became so successful in parallel with an increase in the efficiency of the biological pump over the last 50 millions of years. And with the next slide, I'm going to just take a short break from plankton, because this is a figure from a recent paper which came out a couple of months ago, which I think is very interesting. And it basically shows that the evolution of deep dwelling fishes actually happened in two pulses. And the last of these pulses started basically 15 millions of years ago. So it seems like this evolution of deep dwelling forms is a kind of a widespread behavior in marine organisms. Okay, so now I'm going to move on to the third questions that I wished to address with this talk, specifically what could have been the evolutionary mechanisms behind the evolution of the new deep dwelling species in planktonic foraminifera. Again, this is all based on our fossil data set, and I'm going to give you two case studies as an example. The case study of Hirsutella scitula and of the Hirsutellids, and the case study of Truncorotalia crassiformis and of the origin of the Truncorotallids. I'm going to show you that the origin of the modern Hirsutellids from scitula could have happened through that sympatry, and the origin of Truncorotalia crassiformis from the Hirsutellids and of the Truncorotalids from crassiformis could have happened through a combination of allopatry and depth sympatry. Okay, so let's start with Hirsutella scitula, which is the most ancient deep-dwelling planktonic foraminifera species that we have in our data set. We have it since 15 million of years ago, especially in the Southern Ocean, where it is fairly abundant, about 8%. And it also occurs at site 516, although pretty rare. By 12.5 million years ago, we find Hirsutella scitula also at site U1338, pretty rare as well, but it's rare, it's there. At 10 million years ago, we find Hirsutella scitula at site 872 as well in the tropical Pacific, although it was so rare that I could find it while I was picking for stable isotopes. But then when I was counting for abundance, even though I was using a separate split of sample, I could not find it anymore. So it was there, but really exceedingly rare. However, by 7.5 million years ago, we really find scitula at each of our investigated sites, with abundance ranging from 0.2% to 2.3% in the tropics, and still fairly high abundances in the Southern Ocean. So basically moving from the middle Miocene to the late Miocene, Hirsutella scitula has successfully spread from the subpolar Southern Ocean to the tropics and through the tropics. Okay, and then when it gets to the tropics, apparently it starts doing something else. I've showed you this plot before, but now I want to focus on Hirsutella scitula, which is this green star here. And basically, you can see by the late Miocene, it gets to the tropics, and then the next thing it does is basically moving down the water column, occupying a greater depth habitat through time. And while it is doing that, it is also doing something else. So this is the phylogeny of Hayes et al. 2011. This is the late Miocene, the Pliocene, and the Pleistocene. And as you can see, by the late Miocene, Hirsutella scitula gets to the tropic, starts to sink down the water column, and also it starts to speciate. And we have all these new Hirsutelli species branching off. So what's going on here? Let's have a look to our stable isotope data again to see if we can find some hint. This is again site U1482, southwestern tropical Pacific, 4.5 million years ago. We find Hirsutella margherite, Hirsutella theieri, and Hirsutella scitula. If we look at their oxygen isotope, value, moving from margherite to scitula, we see progressively more positive value. If we look at their reconstructed depth habitat, the shallowest depth habitat is the one occupied by margherite, and the deepest, the one occupied by scitula. In the model sample, we find Hirsuta and in scitula down here in oxygen isotope space with again more positive oxygen isotope value and that deeper depth habitat reconstruction. Site 242, Indian Ocean, 4.5 tropical Indian Ocean, 4.5 million years ago, and we find again margherite and scitula down here in oxygen isotope space with more positive oxygen isotope value and a deeper depth habitat reconstruction compared to that of Hirsutella margherite. Site 516, mid-latitude South Atlantic, 4.5 million years ago. Again, we have margherite up here in oxygen isotope space and scitula down here with more positive oxygen isotope value and a deeper depth habitat compared to that of margherite. Modern sample, Hirsutella hirsuta here, Hirsutella scitula here, again with more positive oxygen isotope value and a deeper depth habitat reconstruction compared to that of Hirsuta. So bottom line, the ancestor species, Hirsutella scitula, always displays a deeper depth habitat than any of the daughter species. So based on this biogeography distribution, abundance data, and paleo-depth habitat reconstruction for both scitula and the daughter species, I tried to come up with an evolutionary model for the modern Hirsutellids. So we just see that by the late Miocene, Hirsutella scitula gets to the tropics and starts to sink down the water column. Meantime, new species start to originate. And here I'm showing Hirsutella margherite just as an example. So what I think what I think is happening here, because we see all the daughter species appearing at shallower depth in the water column, I think that a possible explanation is that the shallower living population of Hirsutella scitula were getting progressively reproductively isolated from deeper living population of Hirsutella scitula, so much that the upper living population could eventually evolve into something else, for instance, Hirsutella margherita. This mechanism is called depth sympatry because the species is actually living in the same area, but with vertically separated water population in the water column. So moving on with time, Hirsutella scitula sinks further down the water column and new species appear at shallower depth. These are the modern Hirsutellis, Hirsutella teyeri and Hirsutella hirsuta, that together with scitula represent the modern Hirsutellis. And based on observation in the modern ocean, these two species, they are still deep dwelling species, but they occupy a shallower depth habitat than Hirsutella scitula. So again, what I think could have happened here is that shallower living population of, for instance, margherita in this case, were getting reproductively isolated from deeper living population, so much that the upper living population was eventually speciating into a new species, originating new species. So then let's move on to Truncorotalia crassiformis and the Truncorotalids. And again, let's start with biogeography and abundance data. So we start having Truncorotalia crassiformis in our data set by 4.5 million years ago, since these species originated in the late Miocene, so we do not have it in our earlier time slice. And it's already quite abundant at site 1138 in the Southern Ocean, again, close to 20%. And is also fairly common at site 516 in the mid-latitude South Atlantic, close to 9%. It also comes with daughter species again as well, such as Truncorotalia oceanica and Truncorotalia confrontatosensis, which is a morphotypes intermediate, to me at least, between crassiformis and tosaensis. So at 4.5 million years ago, we do not find Truncorotalia crassiformis anywhere else at our investigated sites. However, by 2.5 million years ago, we find Truncorotalia crassiformis at each of our investigated sites. It comes with daughter species as well. For instance, at 516, we have Truncorotalia viola. At site 242 and U1482 is a crassiformis occurs with tosaensis and Truncaturinoides. And this is consistent with the Southwest Pacific and Indian Ocean being the area where Truncorotalia truncaturinoides is thought to have first originated and spread out from. And then at all the other investigated sites, Truncorotalia crassiformis occurs either alone or with tosaensis. So again, moving from the early Pliocene to the late Pliocene, Truncorotalia crassiformis appears to have successfully spread from the subpolar Southern Ocean to the tropics. Again, an evolutionary model to try to explain this behavior. Here I'm starting with Hirsutella sibauensis, which according with the phylogeny of Hayes et al. Is the ancestor of Truncorotalia crassiformis. And similar to what I've said before, could have evolved from in the tropical ocean. So it does descend from scitula, but similar to what I've said before, it could have evolved in the tropical ocean through depth sympatry. Through depth sympatry from scitula. Next step, it could have spread towards other latitude, like for instance the subpolar ocean, where subpolar population of scitula could have got reproductively isolated from tropical population of scitula, for instance, to different timing in the reproductive cycle. So much that subpolar population of sibauensis could have eventually evolved into Truncorotalia crassiformis. That maybe could explain why Truncorotalia crassiformis already so abundant in the early Pliocene in our Southern Ocean site, but it hardly occurs anywhere else. But then by the late Pliocene, we found it everywhere in our tropical sites, at our tropical sites. So again, once it gets to the tropics, Truncolotalia crassiformis, it starts doing something else. And similar to Schitula, it starts to occupy progressively deeper depth habitat into the water column. We can see this here. This is the early Pliocene. It is about 300 meters depth here, and then 450 meters in the Pliocene in the Indian Ocean, and 600 meters by the Holocene in the Pacific Ocean. And so again, it moves to the tropics, it starts sinking down the water column. And if we look at the phylogeny of Hayes et al. 2011, it also started to speciate. This is crassiformis. By the middle to late Pliocene, we saw it has spread to the tropics and it starts sinking down the water column. And we see all these new species of Truncolotalids branching off. So again, let's look at the stable isotope to see if we can understand what's going on here. Here I'm showing the data for site U1482, 2.5 million years ago. We have Truncolotalia truncatulinoides, Truncolotalia tosaensis, and Truncolotalia crassiformis. And if we look at the oxygen isotope value, they get progressively more positive, moving from Truncatulinoides to crassiformis. If we look at the depth habitat reconstruction, the shallowest depth habitat is the one of Truncatulinoides, and the deepest depth habitat is the one of crassiformis. Site U1490, 2.5 million years ago, again, Truncolotalia truncatulinoides and Truncolotalia crassiformis with more positive oxygen isotope value and a deeper depth habitat compared with that of Truncatulinoides. The modern sample, we have only crassiformis here with an even deeper depth habitat reconstruction. Site 872, tropical Pacific Ocean, 2.5 million years ago. Again, we find Truncolotalia tosaensis here and Truncolotalia crassiformis. Crassiformis, again, with more positive oxygen isotope value and a deeper depth habitat reconstruction compared to tosaensis. The modern sample, Truncolotalia truncatulinoides and Truncolotalia crassiformis, again, crassiformis with a deeper depth habitat reconstruction. So again, the ancestor species, in this case crassiformis, always displays a deeper depth habitat compared to any of the daughter species. So again, based on biogeography, abundance, and paleo-depth habitat reconstruction, I try to, to reconstruct this evolutionary model. We saw that by the late Pliocene, Truncolotalia crassiformis has spread into the tropics and it starts sinking down the water column. While it sinks down the water column, new species appears. Here I'm showing Truncolotalia tosaensis as an end member, and also because I don't have any of the intermediate morphotypes in my samples. So I think that a shallower living population of crassiformis could have got reproductively isolated from deeper living population of crassiformis, to the point that these shallower living populations were eventually speciated to something else. So through time, crassiformis keeps moving down the water column, and also the new originated species progressively occupy deeper depth habitat. And again, they may got reproductively isolated from shallower living population of the same species. So much the new species were originating, namely Truncolotalia truncatulinoides, which when it first appears in our record, it always occupied a shallower depth habitat compared to that of the other Truncolotalids. Then by the model, we know that tosaensis is extinct already, and Truncolotalia truncatulinoides has in the meantime moved to occupy an even deeper depth habitat in the water column, closer to that of crassiformis. So it really seems like that sympatry, this mechanism, is a very efficient way of generating diversity and inducing speciation among planktonic foraminifera and maybe other plankton as well. And to this purpose, I'm showing in my last slide these very cool results from the Weiner et al. 2012 paper, which I think is a very cool and elegant paper, which shows the distribution in the modern ocean of the genotypes of a modern microperforate deep dwelling species of planktonic foraminifera, Asterigerina pelagica. So basically, Asterigerina pelagica has three different genotypes, genotypes 1 and genotypes 2A and 2B. And these three genotypes, they co-occur in the global ocean. However, genotype 2A and 2B, which are more closely related to each other, they occur very tightly separated in the water column. So genotype 2b always occurs in the upper 100 meters of the water column, while genotype 2a always occurs below 100 meters depth. And if we look at this plot, we also see something else. We see that the genetic distance, the genetic difference between genotype 2b and genotype 2a actually increases with that. So it really seems like that having vertically separated population in the water column is an efficient way to separate the population, keep genetic diversity, and build up genetic diversity, eventually leading possibly to speciation, the origination of new species, the origin of new species, and therefore increase the diversity. Okay, to conclude, what we found is that food, possibly oxygen, and light availability at depth increased with ocean cooling over the past 50 millions of years, making the deep ocean more hospitable for life. This worked as an evolutionary trigger, leading to deep dwelling forms of calcareous plankton and possibly fishes as well, to increase in abundance and diversity on a global scale. Based on our fossil record reconstruction, it seems like that the mechanisms that could have been involved for the origin of new planktonic foraminiferal deep dwelling species were a combination of depth sympatry and allopatry. Allopatry seems to be more important for cladogenesis, so for the branching off of an entirely new group, while depth sympatry seems to be more important for intralineage speciation. And thank you very much for your attention.
Excellent talk, Flavia. Thank you so much.
Thank you so much.
It's astounding the complexity of adaptation to these environments and how depth sympatry possibly works in a dark environment without so obvious connection to lunar phases or seasonal phases and things as strongly as we might assume. And so the details of the separation of the genotypes is a bit of a mystery, just sort of how they actually become reproductively isolated, even though there's overlap in those ranges. But what you show is a beautiful data set that brings a lot of information together. I'm surprised at the diachronaty of first occurrences in some of these things, and so being mostly a Cretaceous worker, this is very enlightening. So thanks very much.
Thank you.
I invite the visitors to ask questions. Just go ahead and raise your hand and then unmute yourself and ask Flavia directly. So are there any questions? Go ahead, Professor Sina. Can you unmute?
Thanks for the very nice talk. I have a couple of questions. One is that as you are dealing with Crassiformis most of the time, that is a deep dweller and its descendant Tosansis and Truncalcnoides. One of the most enigmatic thing which I have observed in the evolution of Truncorotalia is that from Crassiformis we have non-keeled Tosansis, and then again the keeled Truncalcnoides appears, which is there in the recent, while the ancestor Crassiformis and the Truncalcnoides both live in the modern ocean, while the intermediate form Tosansis without keel, it disappeared around 0.6 million years, and it is mostly synchronous in the world ocean. Mm-hmm. So your talk points towards the evolution, the sympatry and allopatry. Can this data set try to answer the question of evolution of keeled to non-keeled and again to keeled, while the non-keeled form disappears?
I honestly don't think so, for the reason that this is a very low resolution study. And I think for what you are saying, you really need high resolution sample where you can really track all these, coming and going of the keels that you mentioned. But with this type of data set, we really have snapshots of time in between with a couple of millions of years in between, you can resolve the details. That's also why, for instance, for my evolutionary models, I'm just showing end members. That's because that's all I got, because I have this low resolution, so I miss all the intermediate forms in betweens. And so I think, to answer your question, I think for what you're for this type of code, you really need high resolution sample and a high resolution data set where we know that those species or intermediate morphospecies can occur, and really try to resolve these changes at high resolution, basically.
Thank you. The last question is, as you told that it is during 15 million years, once you have Antarctic ice sheet developed permanent, and then you have the changes in the deep water ambience with more food availability and more oxygen. Similar thing we should expect at the time of northern hemisphere glaciation, that is around 3 MA, where again there was a major change in the ocean circulation. And we observe major extinction, faunal turnover in planktic foraminifera around 3 million years. That is 3 to 2.8 million years. At that time also, do you see a jump in the increasing depth habitat of these deep dwelling planktons? Just like you observed at 15, do you observe it at again an episode around 3 million years?
Again, we do not have that time interval. We basically have only, no, I'm not sharing anymore. So basically we worked on 7 time slices, and those time slices are 15, 12.5, 10, 7.5, 4.5, 2.5, and the model. So we lack anything in between.
Okay, thank you.
No problem.
Question from Yaron. Go ahead.
Hello, thank you for a very interesting talk. Can you exclude that the change you're reconstructing for the deep dwellers is not simply because of a temperature decrease at a fixed habitat depth where they were living? And as a second part to that, how does this depth separation between the different populations work when a lot of these species, at least in the present day, are migrating through the water column? So they would be mixing anyway, then, I would assume.
Okay, so to answer your question, can I share my screen again? Your first question, I mean. Can you see my screen?
Yes.
Okay, so to answer your question, the depth habitat reconstruction for planktonic foraminifera is based on a combination of stable isotopes and the model. So the model has different configuration depending on the age, and therefore different boundary conditions as well. And it does take into account like background changes in ocean temperature. So any change in the oxygen isotope that we may see that is due to changes in temperature gets corrected in the model, and therefore the depth habitat reconstruction that we obtain for that already takes into account the cooling component. And the change in depth that we see is something extra on top of that, basically, if that makes sense. So it's not based only on the oxygen isotope, where we could have a mixed signal, increasing depth and cooling. It's based on the oxygen isotope profile for from the model, which takes the cooling into account already.
And on what are the temperatures and the cooling in the model being based? Is it based on actual proxy data then, or on what is collected?
It's based on proxy data, and it's based on the model forcing, CO2 forcing. like any other, like any Earth system model works. But I mean, as I said, this is all published. So if you really want to, like, I'm not the one who developed the model, Catherine, as I said, developed the model, and all the details, we have like 2 publications for the only for the modeling, 2 publications only for the modeling to explain all the details, how the model was built, and all the parameterizations and the forcing. And this is one, like geoscience model development, was published last year, and then we have another one that was published this year again. However, the cooling in the model is for sure based on the forcing that we gave to the model, so CO2, and in turn that one was anchored to deep like benthic oxygen isotope data since the middle Miocene, basically. And to answer your second question, that was about how the separation actually occurs between like vertically separated population. I'm not sure if I have an answer to that. To me, is that like a possible explanation is that when populations start to live like significantly distant, vertically distant in the water column, that is probably somehow harder for them to get in touch and mixed with each other. That's my explanation. And I think the last slide where I was showing the genetic data, the genotypes for pelagica, that seems to support this idea that, I mean, the deeper dwelling genotype is getting progressively more different from the shallower dwelling genotypes the deeper you go. So it seems like the more distant they are, the more genetically different they become. How that actually happened, I'm not sure.
So for paleo reconstructions, we may actually have a problem if we want to reconstruct deeper water temperatures, because
yes,
we may have a very clear depth habitat change in there.
Yes, that's also what we say in the science paper, definitely, because we always assume that the forams live at a constant depth, and changes that we see reflects, I don't know, shallowing of the thermocline, changes in stratification, cooling of deeper water, but it can be something else. It can be a different story, like paleoecology story.
Okay, thank you.
No problem.
Is there another question? Yes, Maria Rose.
Oh yes, so Flavia, thank you very much. I really enjoyed your presentation. Very nice, great presentation.
Thank you.
So congratulations. I only have actually a curiosity. It's about Schitula. Actually, I see that basically it is quite abundant at the Kerguelen Plateau. And it first appears at the high latitudes. Is it true? I mean,
yeah, yes. I mean, in our data set, yes, at site 1138, I started finding it at 15 million years ago already. So in the oldest samples that we got, but it was not there anywhere else. And then the more I was working on younger samples, the more I was finding it more and more, basically, in the tropics.
Is there a synchronicity with the record in the Mediterranean, as far as you remember?
I honestly don't know, because I don't know much about the Mediterranean, to be honest.
I don't remember, so I would like to check. So that's very interesting, actually, that they basically are really so common at the Kerguelen Plateau at that paleo latitude compared to the other sites. Quite interesting.
Yeah.
Okay, thank you.
No problem. Thank you, Maria Rosa.
Yeah, and also that it took 7 million years to reach the Pacific Ocean too. I mean, that's to me kind of shocking.
Yeah,
So,
wow. This is wow.
Is there another question? So I'll ask one, since I don't see any in the chat. I wondered, so with this change in the efficiency of the carbon pump, there must be an effect on the benthos, and I'm wondering, has there been a link with the observed extinction of, for example, stylostomelids that Bruce Hayward's discussed, or in terms of the timing, is there a noticeable link with benthic foraminiferal records and a shift in what in their distributions and in diversity and so forth? Has anybody made that connection?
Yeah, I mean, we have some evidence, actually. I mean, this is something we are working on. So there is Chris Smart, who's working on the benthic assemblage data from the same sample I've been working on, but we don't have data yet. However, we just compare this with some older plots that Ellen Thomas produced. And there is, like, we know that since the middle Miocene, basically there is a progressive increase in benthic foraminifera accumulation rates, especially in the low latitudes, if I'm not wrong. And so we did make this link that actually this increase in accumulation rates of benthic foraminifera could also be linked to an increase in the efficiency of the biological pump since the middle Miocene. I think we mentioned that on the science paper as well, I think, if I remember correctly. Yes, so I think that it's there, and we are working on this, on my sample, I mean, on the sample I was working on, to actually develop, you know, same sample data, benthic foraminiferal data from those, but we don't have the data yet.
Excellent, thank you. Is there anybody else with a question? I don't see any. Well, Flavia, thank you so much for an excellent presentation.
Thank you.
Just a reminder, our next speaker will be Rajiv Nijam at 15:30 CET, January 13th. So please join us for our next TMS Cushman Foundation seminar. Thank you for joining everybody, and happy holidays to you all. Okay.
Thank you very much, everyone, and
Merry
happy
Christmas.
holidays. Bye.
Bye.
Thank you.
Bye. Thank you.
Thank you. Bye-bye.
Bye-bye.
Okay, so I have to figure out how to make this recording available. So